We have now to go on to the next topic, and these will be two presentations about the analysis of immune response by functional genomics approaches. And the first speaker will be Joana Bernardes from Kiel. She is a postdoc at the Institute of Clinical Molecular Biology and the research group Systems Immunology, which is headed by Philipp Rosenstiel. And the title of her talk is Single-Cell Transcriptomics and COVID-19 Disease Trajectories. So, Joana, the floor is yours. You are well prepared. I see it. Please go on.
Thank you very much. First of all, I would like to thank the organizers for giving me time to give this talk. As I said, my name is Joana Bernardes, and today I will be presenting this work. This was a very big group working on this specific project, but together with Neha Mishra and Florian Tran and our PI Philipp Rosenstiel, I would like to show you what we have found, and we have recently published. This will be in the end of the presentation. So we are a part of the Decoy Initiative, and specifically because Joachim was talking about the working groups, we are in the working group that is concerning modulation of disease course and response to potential drug candidates. So what we think and what our specialities are is that we are very good in doing kind of longitudinal type of work. So as you know, COVID-19 is one disease, however, it shows multiple disease outcomes. It's very heterogeneous where we're talking about the speed of the disease or the severity. One thing that are in common is if it's severe, you can find acute respiratory distress syndrome, cytokine storms, and this can lead to multi-organ failure. There's a lot of things that we still don't know. We still don't know the long-term disease outcomes, for example. There is no one medication that you give a patient when he comes with severe symptoms to the clinic, and only recently the vaccination has been available. But we still are unaware of this long-term immunity and what it can cause to humans. So what we do know, and there's been a lot of work done, is some cell phenotypes. For example, we have up here on top, we know that there is a dysregulation of the myeloid cell compartment. This is great work done by our colleagues in Bonn. We know that the T cell compartment is also kind of involved, especially in pre-existing antigen-specific T cells. We know, and a lot of studies have shown, that there is an expansion of plasmablasts or plasma B cell compartment that is specific to COVID-19. And also some of these plasmablasts seems to show kind of neutrophil-like phenotype. We're still not sure what the natural killer or NK cells are doing throughout the disease, but one thing pathologists have been identifying is that with this disease, you can find kind of the patients that unfortunately pass away from this disease, so 95% of them show immunothrombotic events. And this happens when you have a platelet and megakaryocyte rich environment within the patients. This is more than you can see in typical infectious disease, and it can also affect other organs besides the lungs. So what our project focuses on is doing a longitudinal sampling and multi-omics analysis. When I talk about multi-omics, what we are talking about here is bulk RNA-seq and bulk DNA-seq that can lead to methylation information. And we have single-cell sequencing. In this case, we did 5' with BCR and TCR repertoire together with flow cytometry. We also took serum samples to do multiplex analysis and so on. So a lot of omics layers that we try to work with, and our study had, it was particularly in the case that there were 2 centers. So we had samples coming from the University Hospital of Kiel and Cologne, and dense clinical phenotyping. Also, from this project, we were lucky enough to get our hands on the data from the paper from Joachim's group. And we use this as kind of a validation cohort for our findings in our own cohort 1. So I talk about patient trajectory, and what I mean by that is also pseudotypes throughout the paper. So what we do is for each individual each individual sample of one patient, we identify the WHO ordinal scale and also different inflammatory markers such as CRP, IL-6, and ferritin. And these all work towards a compound score that goes from 1 to 7. So at 0 pseudo time is our control samples, which is healthy samples, which will be depicted as gray throughout the presentation. And then we have kind of a color scale and a numeric scale that goes from 1 to 7. So at 1, you have kind of incremental. So the patient is in ascending, getting worse part of the disease. Critical is when the patient is so it's heavily ventilated, the inflammatory markers are going very, very high. And then from this on, it could be that unfortunately the patient passes away or the patient recovers, and then you have still a hyperinflammatory phase, and then you have recurrent and convalescent state. We were lucky enough to get samples from patients that had a long-term follow-up, so they do not show symptoms for more than 6 weeks. And this was all integrated in our project. So the first thing we did using the single-cell transcriptomic analysis was to merge all the samples. And here you can see in this UMAP, we had over 350,000 cells and their transcription information. So here you can see that the PBMCs, you can identify monocytes, CD4 T cells on orange, CD8 T cells, NK cells, and then other specific clusters you can see up here in green is the megakaryocyte compartment, and below you find in this brown color plasmablasts, and in yellow the B cells. So what we did after was kind of to paint the cells using the pseudotypes, and we could already infer some differentiation between our pseudotypes, especially in the monocytes. But to have an easy evaluation of how the cell types change throughout the pseudotypes, I plotted here this graph. So in gray, you have the healthy samples, and then you have the incremental, so inflammatory phase of the disease in pink and purple colors, and then you have the convalescent state, and blue is what we call recover patients. So we could indeed identify what other people have identified before. There was increase in the number of cells belonging to the monocytic compartment. There was lymphopenia. You can see here a decrease of the CD4 T cells in comparison with the healthy control. You can see that the percentage of plasmablasts really increased, so throughout the disease in comparison to the healthy control. And the megakaryocytes here, the last graph on the bottom right, you can see that there is a difference on the number of megakaryocytes throughout the disease. So this pseudotime is indicating something. So going further into the megakaryocytes, what we found is that if you see here the center panel, that in the megakaryocyte cluster, there seems to be a localization that changes throughout the disease. So here on top, you have the healthy cells, and then you have the incremental phase, and from here on, you have this critical phase, which is really has quite a differentiated transcription profile than the other cells. And this is first here and comes and disappears. And then you can see that in convalescence and in recovery stages, the megakaryocytes transcription profile is much more similar to the healthy controls that we had. One thing that we can also, excuse me. Excuse me. So one thing that we could identify is that the megakaryocytes response have interferon signaling, and this interferon is throughout the inflammatory stages. So if we focus here on IFID-M3, for example, you can see that incremental and critical, there is a great expression of this gene during these phases. Also, below here, you can see this is cohort 2, that we could also confirm this from on. So once you kind of identify the signature genes for the pseudotypes, you can also do top geo enrichment analysis. This is what we did. And of course, we saw something that was a little bit unexpected. That was that the cellular type 1 interferon was upregulated throughout the course of disease. And then we have more specific terms such as platelet aggregation and platelet degranulation that are kind of characteristic of having a very critical, during a very critical phase of the disease. Passing on to the B cell compartments, we also, as I said before, identify an increase in the plasmablasts. You can see here on the center U map. So on the left, you have the memory B cells and naive B cells. And then on the right up top, you have the plasmablast clusters. And you can see that they have different colors. This is because I overlaid so I put on top the cells that have a healthy, that are from the healthy samples. And here you can see that there is a lot of memory B cells from the healthy donors, but not so much if we look at the plasmablasts. We also work close together with some colleagues from Kiel, from Christoph Kaleta's group, and they do wonderful metabolic modeling. And here on the right, you can see that we found a lot of different types of metabolism, special catabolism, that was highly activated throughout disease. When in the plasmablast specifically, once we compare it with a control. And we advanced that this might be serving as a nutrient sink, which is kind of a model that was already reported in malaria diseases. So the plasmablasts are very metabolically active. Going further on the plasmablasts during disease, we also identify neutrophil-like cells in the next to the plasmablast cluster. However, you can see that these neutrophil-like cells were also present in the control, so not so specific to the disease. And what we found was a sustained upregulation of unfolded protein and mitochondrial ATP synthesis. Here depicted, for example, you see this XBP1, which is a gene related to unfolded protein, highly expressed in the plasmablasts, not so much in the other cell types. And you can see that is very increased during disease. While you also have here a gene related to the mitochondrial ATP synthesis, and this is also upregulated throughout disease. And finally, I would like to just give a quick look on the monocytic signatures. This is more like a confirmation of what is already seen in the paper from Joaquin's group. So we do find the same increase in classical and non-classical monocytes. So non-classical being CD16 positive monocytes. And we can also confirm this HLA-DR suppression during very critical phases, so up here, especially in the classical monocytes. And here also critical is very different from the adjacent pseudotypes. One of the great perks of our work is that we had multiple omics layers, and we also had 2 cohorts to validate our findings. And this is just to finalize this presentation. I would like to show you this validation. We use this cohort from Netherlands. And as you can see, we focus here on the middle panel. We have megakaryocytes co-expression models. So these co-expression models were identifying the bulk RNA sequencing, and these were the most related to the megakaryocyte proportion and expression profile. And this kind of gives us some biomarkers that can help us identify very nefarious disease predictions, especially for the non-survivors of the cohorts. So the take-home message from our project is we did this comprehensive view at cellular dynamics. We could confirm this expanded plasmablasts, and we advanced that this might be related to this nutrient sink theory within the immunometabolism. We found emerging megakaryopoiesis, so there was an increase of the megakaryocytes count on the peripheral blood, and this could be indicated that this is what's happening. There is an activation of platelets that can lead to of thrombotic events. And finally, we corroborated a sustained increase and shift in the monocytic compartment. If you want to read more about our work, I would like to refer to you to our paper. It's called Longitudinal Multiomics Analysis. And if you do this, you can find COVID-19. This is, you can find us there. These are all the people that contribute to our work. You can see there's a lot, most of them from the Decoy Initiative. And also, if you're specifically interested in the single cell analysis, you can go to Fast Genomics and put the name of our paper, and you can literally go and check every single cell from our paper in a very interactive manner, which is really, really cool if you're interested in the single cell analysis. And yeah, I would like to finish by thanking my PI, Philip Rosenstiel, also Rakim, for creating this environment that help us communicate so much better and creating fast and kind of fast and furious results that are relevant for the climate that we are living right now. And thank you all for listening, and I will now get questions if you have them. Thank you.
Joana, thank you very much for presenting this impressive work. So I have not received so far a question from the audience, but I would like to know from you, once you understand all the developments of the immune cells in the patients and the differences, how can you translate it into a better therapy?
Yeah, so some of the patients of our cohorts were actually treated with remdesivir, for example. Unfortunately, this is very difficult. It was very difficult for us to identify target engagement of the therapy. This is normally what we do. First, we see if the therapy is working or not. And I think if we increase the number of patients in our cohort, and if the therapeutic is actually working, is better to kind of map what the therapy is doing, and then if the therapy is doing what it's supposed to. So if the patients are getting into remission early on or not. We are still not there. I think we need more patients, and we need better and newer therapeutics to figure it out what's going on.
Okay, thank you.
Thank you.
So, is there